Hello. Hello. Can you guys hear me? Hello.
Hello.
Okay,
Yes,
that's great. Okay. So yesterday we discussed about immunology and we discussed the innate immune system. We discussed adaptive immunity. So today we are going to discuss, a little bit in detail, right? But before starting today's lecture, let's first of all summarize what we learned yesterday, right? So I just told you about the concept of antigen presenting cells and everything. So who is going to be giving me a quick revision about that? The extracellular pathogen, all of the CD8 positive cells. We discuss each and everything in detail. The MHC2, the MHC1, and everything. So who is going to be giving me a quick overview of that? Hello, anyone? Hello. Yes,
hello.
please. Yes, please.
Okay, so there are antigen presenting cells. The antigen presenting cells are basically the cells that present the antigen
Mm-hmm.
to the T cells. And
Okay.
there are four types. There are B cells, dendritic cells, macrophages, and the Langerhans cells.
Okay. Perfect. Okay.
So if we have an exogenous antigen that is presented to the MHC type 2 cell, and
Okay.
the CD4
Mhm.
activated in that case.
Perfect. Perfect.
And they further, and then depending on the requirement, they further activate either Th1 cells or the Th2 cells. The Th1 cells bring the macrophages, and the Th2 cells bring the antibodies.
Perfect. It means that the yesterday's sessions have helped you guys a lot. You guys know everything, right? So let me summarize it again and then we are going to move. Okay. So we discussed about MHC1. What we discussed that MHC1 is going to present on all of the nucleated cells. If we have any kind of intracellular infection, for example, a viral infection, so what your nucleated cells are going to do, they are going to break that virus into pieces. They are going to present it on MHC class 1, right? Okay. Then your CD8 positive cells are going to come there and they're going to eat the cell, assuming that we already have virus, which is being presented by MHC class 1. Is that clear? Hello?
Yes.
Okay. We also discussed another concept. Sometimes your viruses won't allow the presentation of MHC1 on the surface, right? It's a clever mechanism for the viruses to hide from your inflammatory cells or your cytotoxic CD8 positive cells. So in that case, your natural killer cells are going to destroy those cells who do not have MHC1. Is that clear? And for the extracellular pathogens, we discussed that antigen presenting cells are going to come. They're going to eat the extracellular pathogen, present it on MHC class 2. 2 into 2 is 4. They're going to present those to your CD4 helper T cells. And we know that CD4 helper T cells are further divided into two types, Th1 and Th2. So Th1, T helper 1 cells, T helper 2 cells. The T helper 1 cells are gonna, they're gonna recruit your macrophages. Okay, so we are going to discuss in detail the interleukin 12, the interferons, alpha, gamma, and everything, right? So it's not the time to discuss that, but you should know that there are some molecular messengers which keeps on activated or keeps on, connecting different cells with one another. Those molecular messengers, cytokines, and interleukins. We're going to discuss that in detail. Okay, so we discuss these things. Okay, so let's discuss MHC1 and MHC2. Remember, there are something called HLA. What are HLA? That's very simple. HLAs are basically the cluster of genes which basically encode for MHC. That's all. That's simple, right? Okay. HLAs or human leukocyte antigens are cluster of genes which encode for MHC, right? So after LHA, you can see in the first stage, whenever we have a single letter A, B, and C. After HLA, we have single letters, letter A, letter B, letter C. So the single letter HLA are going to encode your MHC1. MHC1 have one in its name, one for a single letter, right? So MHC1 are going to encode for the single letter HLAs are going to encode for MHC1. While the double letters, if you see the two letters after HLA like DP, DQ, DR. So they are going to encode for MHC2 because in MHC2 name you have the word 2, right? So 2 encodes, the double letter encodes for MHC2, the single letter encodes for MHC1. That's really important, really high. You guys are going to see MCQs on that related to that as well. But I have just made it really simplified for you guys, right? Okay. We discussed that MHC1 is going to be present on all the nucleated cells. Okay. I'm just revising these things again and again in order to, because these are the core concepts of immunology, right? So let's have another revision, right? We have our nucleated cells. On the surface of that, we have MHC class 1. If we have intracellular viral infection, cells are going to break down into pieces, present it on MHC1. And it's basically the goal of your CD8 positive T cells, the cytotoxic T cells, to come and destroy the entire cell. Is that clear? Okay. While in case of MHC2, we know MHC2 are present on antigen presenting cells. MHC2 are present on antigen presenting cells. What can be your antigen presenting cells? B cells can be your antigen presenting cells. Macrophages can be your antigen presenting cells. The dendritic cells can be your antigen presenting cells. The Langerhans cells can be your antigen presenting cells. They are going to take the antigen, present it on MHC2. 2 into 2 is 4. They're going to present all of these antigen complex to your CD4 helper T cells. Is that clear? So CD4 for MSC2, 2 into 2 is 4, right? Is that clear? Hello?
Yes, sir.
Okay, perfect. So okay. What's the function? We already know the function. Intracellular viral CD8 positive T cells, exogenous antigen bacterial protein, antigen presenting cell, MSC class 2, CD4 positive helper cells. We know that. Let's discuss the structure of these, right? Let's discuss the structure of your MHC1 and MHC2, right? So here in the first aid, they have mentioned it really clearly and really simply, right? So you have two chain, alpha 2 chain and alpha 3 chain, alpha 1 chain. So the structure of MSC1 is really important because you're going to see a lot of MCQs, right? And I'm going to mention that MCQ. So you're going to be having one MCQ in your hematology section, one in immunology section, and one in MSK section, right? In your U world, and they're going to ask you the same concept, right? So you're going to have 4 to 5 MCQs only, based on the structure of MHC1. So let me just clarify that. In this structure, can you just see something called beta 2 microglobulin here?
Yes.
Okay. So you can see that in the MHC1, you have something called beta 2 microglobulin, right? Remember, those individuals who are on dialysis, those individuals who are on dialysis, sometimes what happens as this beta 2 microglobulin in MHC1 structure, right? These are present on all nucleated cells. We know that, right? So in dialysis, as these beta 2 microglobulin once detaches from MHC1, come into the blood, they cannot be filtered out through dialysis. They cannot be filtered out through dialysis. So what happens? They are going to build in your body. And once they build in the body, what's happened? They are going to deposit in different tissues inside your body. So that's basically amyloidosis. We're going to discuss amyloidosis in detail, but let me just make it really simpler for you. What is amyloidosis? Deposition of amyloid. What is amyloid? Amyloid is basically, a disorganized kind of proteins. Disorganized kind of protein. We are going to discuss the beta pleated sheath and everything, right? But that's not now, right? But for now, you can just imagine that amyloid is simply a disorganized kind of proteins, right? So the beta 2 microglobulin, they can be amyloid. They can be your disorganized kind of proteins. So in the patient with dialysis, the beta 2 microglobulin is going to build up and it's going to deposit in different tissues, giving you amyloidosis. In that case, we are also going to call it as dialysis-induced amyloidosis, right? And that's basically due to beta 2 microglobulin deposition in different structures. Is that clear? Yes or no? Right?
Yes.
Okay, so this was one of your MCQ. They are going to tell you we have an individual. He was on dialysis. Suddenly he developed an amyloidosis. Okay, and you guys know how do we see amyloidosis from your renal section? Who's going to be telling me plain polarized light? Everything I just told you, right? Apple, something like that. Hello, anyone?
Yeah, apple green bifringence in your Congo red.
Perfect. Yes, whenever we pass the plane polarized light through the amyloid, right? So it's going to give you apple green birefringence once it's stained with Congo red, right? So that's all. So they're going to tell you we have an individual, he was on dialysis. Keep that in mind. That's going to be the key point in your MCQ to diagnose this condition. He was on dialysis. He developed amyloidosis, right? Unlike they're going to tell you we just took a specimen, right? And in the specimen, what happens? That he was having some amyloid. What do you think this the amyloid was basically, structural component of what? It was a structural component of your MHC1. Is it clear? Yes or no?
Yes.
Yes.
Perfect. What else in MSK they are going to ask you? Sometimes this beta 2 microglobulin, these amyloid can sometimes deposit in your, in the flexor retinaculum, this part of your hand, right? And once it deposits there, it can cause something called carpal tunnel syndrome, right? If you have a lot of amyloid there in the flexor retinaculum, it can just build there. It can cause carpal tunnel syndrome. In carpal tunnel syndrome, which nerve is going to be compressed? Yes, again, one of your UL MCQ.
Median.
Median
Median nerve,
Median
nerve.
right?
nerve.
What's the function of median nerve? It's going to supply your thumb, your index finger, and half of, your middle finger and half of the other finger. It's going to be sensory sensation, your thenar muscles, right? The two lumbricals and everything. We're going to discuss that in in our MSK section, right? Innervations and everything. Just don't worry about that, right? So they're going to ask you. We were having an individual. He was on dialysis again. He developed carpal tunnel syndrome. What do you think? What's the cause of carpal tunnel syndrome? Who's going to be telling me that? Yes. Hello.
Accumulation of beta to microglobulin.
Accumulation for
Accumulation of beta 2 microglobulin. Perfect. So you guys can see you cannot ignore the structure of the first aid as well. Right? In the MHC, a lot of students should just go through it. Yes, it's the structure alpha and beta 2 microglobulin. So it's really important to know each and everything and the association between the structural component and the beta 2 microglobulin as well. That's really important. Okay. So let's discuss our next topic, that are different HLAs, right? So we are going to discuss these HLAs, the DR3, DR4, B57, everyone thing. Once we discuss each and every diseases, right? So as we already have discussed diabetes type 1, what was diabetes type 1? Juvenile diabetes, yes. Remember?
Diabetic ketoacidosis.
Mm-hmm. Okay,
It
let's discuss
contains
diabetes in a little bit detail because you guys have done your endo. Let's just do some
HLA D3
MCQs of that.
and HLA D4.
D3 and D4. D3 is also associated with Hashimoto's, remember?
Yes,
Okay.
yes.
So let's just discuss diabetes in a little bit detail, right? Let me just ask you some questions because you guys already have done your diabetes. So let me ask you something related to DM1. Okay. So let me just ask you some simple kind of MCQs, right, in order to test your concepts. Diabetes type 1 is going to be seen in younger individual or older individual.
Younger individual.
Okay,
Less than
perfect.
50.
What's the cause of DM, diabetes mellitus 1,
autoimmune,
type 1?
autoimmune,
Insulin
Autoimmune
deficiency.
insulin
destruction. Perfect.
Abdomen destruction of
So in the in
use of
the in the
Langerhans.
islet of Langerhans, we are going to be seeing leukocytic infiltration or lymphocytic infiltration. Yes. One of your
lymphocyte,
lympho,
Leuco-
lympho,
leucos- Leukocytic infiltration.
leukocytic. One of your UL MCQ. I think you guys haven't did
Yes.
your just concentrate. You have to differentiate what is leukocytic and what is lymphocytic as well. Okay. Okay. What is the complication of diabetes mellitus?
DKA,
Type
DKA,
1.
DKA.
diabetic ketoacidosis.
Diabetic ketoacidosis. Okay, let's discuss some renal concepts as well. It would cause which kind of acidosis? Anion gap or non-anion gap?
Anion gap.
High NN gap.
Anion gap.
NN gap.
Right.
Perfect. Perfect.
High
You guys are doing
anion
really
gap.
great. High anion gap because those ketones can just give you some extra anion if you just calculate it with the formula, taking all of the Na, subtracting from all of the negative ions. If the value is equal to 12 or more than 12, that's an anion gap, right? So these kind of Addison disease. What is Addison disease, by the way? Anyone?
autoimmune destruction of
Autoimmune destruction of adrenal
No, no,
glands.
adrenal insufficiency.
Adrenal insufficiency.
Yeah.
Yes.
Okay.
Primary adrenal
That
autoimmune destruction. Yeah.
Okay.
that could be
So,
because of the in developing countries and in developed countries. So it could be autoimmune and also
autoimmune.
because
Autoimmune
TB.
in developed countries
I-
and
In and in
And TB.
developing countries because of TB.
TB under development countries, they are going to be due to TB. That's really important to know. Okay. Okay. Perfect. Okay. So let's discuss our next topic. Okay, the natural killer cells. We already know what are natural killer cells, what are the function of natural killer cells. So he's going to be telling me that. We already have discussed it in detail. Yes, they are the component of innate immunity, immune or adaptive immune system.
They're
Innate
innate,
immune system.
but they come from
Perfect.
a lymphoid lineage.
Perfect.
Innate immune system.
Okay. So basically, how does these natural killer cells lead to destruction? As we already discussed, whenever the cells do not have MHC class 1 on their surface, your natural killer cells are going to kill them. What are their mode of killing? The mode of killing is that they are going to release something called porphyrins, okay, or porphyrins and granzymes. So as the name
Granzymes.
indicates, what porphyrin would do, it would make pores within the cells, right? It would make pores within the cells. And what granzymes are going to do, granzymes are going to enter, those are the enzymes which are going to enter through those pores and lead to destruction of, virally infected cells or tumor cells. Really important, right? Is it clear? Okay, so we are
Yes.
going to be discussing these interleukins in detail: interleukin 2, interleukin 12, INF gamma, INF beta. Okay, so let's just leave it to that. Okay, so interleukin 2, interleukin 12, INF gamma, just keep it in mind. We are going to discuss each and every interleukin, their function, and everything, right? It's going to be a topic for tomorrow's discussion. Okay. So the next one is the major function of B and T cells. We already know. We already know that. Yesterday we discussed, right? Okay. B cells. B cells are going to make plasma cells. Plasma cells are going to make antibodies. What are the first antibodies made by your B cells? Yes, anyone? Yesterday we discussed
IgM.
it.
IgM.
IgM,
IgM.
perfect.
Yeah.
Okay, if I want some other antibodies, IgA, IgED, then I'm going to do a process or what's the name of that process?
Class
Class
switching.
switching.
Class
Class
switching.
switching. Perfect. I have to switch the class. So how do I undergo class switching? CD40, CD40 ligand interaction. We are going to discuss that, but not now, right? Okay. Okay. So T cells. We discussed T cells. We have two types of T cells: the helper and the killer. Helper are your CD4 positive. Killers are your CD8 positive T cells, right? And the helpers further can be divided into two types: Th1, Th2. Th1, Th2. We know that Th1 is going to recruit your macrophages, while Th2 is going to recruit your B cells. Yesterday we discussed the examples of that as well: asthma, we discussed granulomas, we discussed sarcoidosis, we discussed whether it's a Th1 response, whether it's a Th2 response, right? So we know that. Okay. Okay. In type 4 hypersensitivity, we are going to be okay. Dr. Moaz did type 4 hypersensitivities, right? Hello? He did
No,
No.
hypersensitivities, yeah?
we did type
No.
1, 2, and 3, and he left type 4 for later.
Okay, so we are going to discuss it in hypersensitivity section. So hypersensitivity type 4 basically involves, the T cells. We are going to discuss that. And we're going to discuss the entire, examples of that, urushiol and everything, right? And, so just let's leave it to that, okay? So the next one is, if yesterday you guys remember, I just told you about the primary and secondary lymphoid organs, remember? Hello?
Yes. Yes. Yes.
Okay, okay. So in the primary lymphoid organs, I just told you we have our bone marrow and then bone marrow and what else? What else? Bone
Thymus.
marrow and thymus. Perfect.
Thymus.
We discussed that B cells are made within the bone marrow and they are matured within the bone marrow, while T cells are made within the bone marrow while they get mature in the thymus, right? So how do they get mature? Now let's discuss that. Okay. So I'm going to give you a really great example for that, but I'm going to make a story for you guys, right? Okay, but before that, okay, can you guys just see this thymus?
Yes.
Okay, okay, now it's going to be more clear, right? Remember, whenever the T cells are released from your bone marrow, they are going to be double positive. What do I mean by double positive? They are going to be having both the receptor, both CD4 and CD8 receptor on their surface. Is it clear? Once they are released from the bone marrow, they come to the thymus for maturation process. They are going to be double positive. What do we mean by double positive? they're going to be having both your, they're going to be having both
CD4 and
CD4
CD8.
positive, CD8 positive. One of your UWorld MCQ, right? That's going to be one of your UWorld MCQ. They're going to be double positive once before they have undergone this maturity process, right? Okay. Then they are going to enter into the cortex of your, they're going to enter into the cortex of your thymus, right?
Thymus.
And there we are going to be having something called positive selection. Okay. So you know what is positivity? Positivity is love. Love is positivity, right? If you're loving someone, if you have more attraction, that's basically a positive thing, right? So remember, in positive selection, we are going to spare those B cells and T cells who show love. Is it clear? In the positive selection, we are going to spare those T cells who show love. Love towards what thing? Love towards who? Right? We know that, okay, we know that your MHC2 love your CD4 positive cells. We know that, right? CD4 positive cells love MHC2. We know
Yes.
that, right?
Yes.
Hello?
Yes, we know that.
Okay.
Yes.
We know that MHC1 love your CD8 positive T cells. We know that, right?
Yes.
Okay. Okay. So if your CD8 positive T cells are showing strong love to MHC1, we are going to leave them. If your CD4 positive T cells are showing more love towards the MHC2, we are going to spare their lives, right? That's what we need. That's what we need. It's a positive selection. We need positivity. We need love, right? Hello? Is it clear?
Yes.
So
Yes.
this this love process is going to be passed whenever your MSC2 show love towards CD4 positive, and whenever your MSC1 show love towards your, okay, MSC1 show love towards CD8 positive. Then then we have our next process, that is negative selection. So what do we mean by negative selection? Whoever is showing love, we are going to kill them. It's a negative thing. Whoever is showing love, we are going to kill them. Positive selection, whoever is showing love, we are going to leave them. In the negative selection, whoever is showing love, we are going to kill them. But now the question should arise in your mind: love towards what thing? Love towards, what? Okay. So remember, here in our body, we have something called, we have a company, right? We have a company, a car company. That's the company. The name of that company is ARE, AIRE, right? Okay, just listen the story, right? In our body, we have a company named AIRE, ARE company who make cars. ARE company who make cars, right? And what's the function of those cars? Those cars are going to take antigen from different parts of our body. Like they are going to take some of the, some of molecules from your heart, some of molecules from your liver, some of molecules from your kidney, some of molecules from the pancreas, and they are going to load it into this car, and they are going to take it into your the region where your negative selection is occurring. Is it cleared? Yes or no? Okay, we have a company who make cars. The name of that company is AIRY. Okay, it would make car, and what those cars are going to do, they are going to take antigen from all of your body parts. They are going to load it into that car, and they are going to take it into the zone of thymus where negative selection is occurring. Is that clear?
Yes.
Okay. Okay. And now, if the T cells are showing love towards your own body tissues, which are being loaded in the car, we are going to kill them. Why? Because we do not need T cells to show love towards our own body parts. If they show love to our own body parts, they are going to kill us. So, negative selection is a good process for us, right? Although we are killing those who are showing love, but they are showing love to different, to wrong people. No, so whoever is showing love to the wrong people, we are going to kill them, right? Is it clear? Hello?
Yes.
Okay.
Yes.
Okay, so let me just ask you a question. For example, if due to any reasons your company, the ARE company which was just making the cars, due to any reason, if it, bankrupted, it get bankrupted and it's closed. So now do you have the cars? Do you have the cars who are going to take things and put it into the negative selection zone? No, right?
No.
So now you cannot test your T cells for your own body parts. Can I say that?
Yes.
Right?
Yes.
So now you have chances that you can get certain T cells which can lead to autoimmunity. Is that clear? Yes or no?
Yes.
So can I say whenever the ARI company get bankrupted, whenever the ARI company get bankrupted, we are going to be having problems because the T cells are going to kill us then, right? Is it clear?
Yes, it will
Yes
be
or no?
autoimmune disease.
That would be autoimmune disease. And this process is also called as central tolerance. Is it clear? Central tolerance. Right?
Yes.
Okay. Any questions? Any questions?
No.
Okay. I think the positive and negative selection is now quite clear, right? Positive selection, whoever is showing love, they're showing love towards right people, just leave them. Negative selection, they are showing love towards wrong people, just kill them. That's simple, right? We have a company called ARI, right? ARI company. So let's just, summarize. Let's just read our first aid. Okay, so I'll be reading the first aid and you guys are going to imagine the entire story, right? Okay, the thymus cortex keep T cells that recognize self-peptide to allow for cooperation in immune responses. Double positive thymocytes expressing TCR, MHC complexes, receive a survival signal. I think that, I've explained quite well in first aid, right? Okay.
Yes.
So here you can see the ARI company, okay, the autoimmune regulator or autoimmune regulator protein, that was the ARI company which I was just talking, right? So if we do not have that, we are going to be having some autoimmune condition. What would be those autoimmune condition? Chronic mucocutaneous candidiasis, hypoparathyroidism, adrenal insufficiency, recurrent candida infections, and the mnemonic for that is CHAR, CHAR. Right? So you can just memorize it, right? Okay. Is it clear?
Yes.
Okay, perfect. So let's discuss our next topic, that is macrophage lymphocyte interaction. Okay. So before discussing that, let's again come towards our classical story, which we keep on repeating again and again. We have exogenous antigen. It's going to be taken by your antigen presenting cell, right? Hello. Antigen
Yes.
presenting cells are going to present it on MSC one or two.
One.
One.
Mhm.
MHC2.
Two.
Two.
Also.
You know,
MSH2.
it's being like, I think 50 at time. I'm just repeating the same story again and again, and you guys are saying it's one.
It's one.
Okay, it's not one.
No, it's
Okay.
two.
It's two.
Antigen presenting cells.
Exogenous or on.
That's two, right? Hello, that's two.
Okay.
Okay,
Yes.
and I just told
Yes,
you that
it's two.
antigen presenting cells can be your B cells. Am I right?
Can
Hello.
be.
Yes.
B cells can be your antigen presenting cells. So what B
Yes.
cells would do, sometimes they would just engulf an antigen, present it on MHC class 2, 2 into 2, 4. It would just come towards CD4 positive T cells, your helper cells. They're going to interact with one another, right? And we note whenever the B cells are activated in this way, the B cells, the Th2 response, we're going to be having plasma cells. Plasma cells have the ability to make IgM. But if we need some other kind of antibodies, so we are going to do something called class switching. Class switching. We discussed it, right?
Yes.
Okay.
Yes.
Yes.
So remember, for the class switching, we also need another receptor to come in communication with one another. And what's the name of that receptors and the ligand? CD40 receptor with CD40 ligand. CD40 receptor with CD40 ligand. So CD40 ligand is going to be present on the with cells, T cells, right? And CD40 is going to be present on macrophages. It can be macrophages because macrophages can be your antigen presenting cells, right? B cells can be your
Yes.
antigen presenting cells, right? So you don't have to confuse yourself, right? If here we have written the dendritic cells, again the story is same, right? So in order to undergo class switching, in order to switch the classes, if I need IgA, if I need IgE, if I need IgD, it means that I should be interacting some extra things, some extra receptor, that is CD40, CD40 ligand. Is it clear? Hello?
Yes.
And
Yes.
once the interaction of CD40 occurs with CD40 ligand, then we are going to be having our class switching. Then we can get IgA if we want. And our mucosal immunity, we discussed that. Then we can get IgE. If you guys remember in our asthma, sensitization of your mast cells, degranulation, histamine release, everything. Okay. So, okay, so let me just ask you a question. Let me just clear one of the pathology here. For example, if we have any failure of CD40 ligand interaction, so can we undergo class switching? Yes or no?
No.
No.
If we cannot
No.
undergo class switching, can I say that now only IgM can be made, nothing else?
Yes.
Yes.
Hyper
So
IgM
let me just
syndrome.
name
Yeah.
this pathology as hyper IgM syndrome.
Yes.
You guys have done one of the pathology here, right? Congratulations. Okay. So let's discuss our cytotoxic T cells, right? We discussed what are your cytotoxic T cells. You have helper cells, killer cells. Killer cells, CD8 positive, cytotoxic cells. We know that cytotoxic cells love your MHC1. Every nucleated cells, virus being
Yes.
presented on MHC1, killing everything. Yeah, right? Is that clear? Okay,
Yes.
perfect. Okay, so the next one is regulatory T cells. So we have something called regulatory T cells. What do we mean by regulatory T cells, right? They're going to modulate the activity of your immune system. What do I mean by that? Whenever, okay, they are going to sit there, they are going to see whether immunity is going well. They're going to, see either if we have exaggerated response of CD4 positive and CD8 positive, these regulatory cells are going to come, they are going to suppress them because we do not want anything to get exaggerated, right? Okay.
Yes.
Hello. You do not need something or some process to get exaggerated, right? So what are your regulatory T cells? Just keep that in mind. What are the surface receptors for that? CD3, CD4, CD25, CD3, CD4, CD25, right? And FOXP3. Okay, so okay, let me just clarify it. Let me just give you a general concept in order to know whether okay you are going to be having a lot of MCQs in your U world based on this. So knowing this general principle, you can just mark it correct. The low number receptors like CD3, CD4, CD1, CD2, let's suppose, right? They're going to be the surface receptor for your T cells. And the higher number receptors are going to be the surface receptors for your B cells,
B
right?
cell.
CD21, CD20, all of them are B cells, right? Okay. So the lower number receptors for T cells, the higher number receptors for B cells. That's a general concept which you should keep in mind. So, if you do not know even which receptors are present on these regulatory cells, in the option you get CD3, CD4, CD25 is the only exception. Keep that in mind. Okay. So, okay. So we are going to discuss this condition IPEX, but not now. Okay. The endocrinopathy, everything dermatitis. So it's associated with a lot of systems. But we are going to discuss in the end, once we just discuss the entire concepts of our immunology, I'm going to come back to this topic, not now. Okay. So here again, you have the same story. The same story. Who is going to be repeating it again? I just repeated 50 times. So who is going to be repeated for the 51st time? Same story. Yes.
Sir, can you please repeat because I cannot attend.
Okay. So let me just tell you the B cells and T cells activation again, right? Okay. We discussed we have intracellular pathogen and we have extracellular pathogen. What do we mean by intracellular pathogen? Okay, for the pathogen, I mean any kind of antigen, foreign body, right? So, okay, so certain pathogens can be intracellular. For example, viruses, some bacteria can also be intracellular. We are going to discuss that, right? Okay. So, whenever we have intracellular or viruses within the cells, all of your nucleated cells have MHC1 on their surface, right? So, what your cells are going to do, they are going to break this virus into pieces. They are going to present it on the surface MHC class 1. Is it clear? And your CD8 positive T cells are going to come. They're going to eat the entire cell because we know in the positive selection that CD8 positive T cells loves your MHC1, right?
Everyone.
Okay, perfect. Now, what if we have an extracellular or exogenous pathogens that are present outside your cells? For that, we have a system of antigen presenting cells. So, what are antigen presenting cells? They are the cells that are going to come. They're going to eat the exogenous, bacteria. They're going to cut it into pieces, break it down into pieces. They're going to present it on MHC class 2,
True.
right? MSC class 2. Once they present out on MSC class 2, after that, their 2 into 2 is 4. They're going to present these
CD4.
pathogens or these particles to CD4 helper cells. Is it clear? Okay.
Yes.
Once they present it to CD4 positive T cells, we have two pathways, right? TH1, TH2, TH1, TH2. If we need macrophages to get recruited towards the site of infection, we need TH1 response. For example, any kind of granuloma, all of the granuloma have your macrophages. Sarcoidosis have non-caseating granulomas. TB have granulomas. So if we not need a granuloma metas inflammation, TH1 is going to be activated. Then we have TH2 response. For that, we have to recruit our B cells. In the B cells response, we already know the antibodies response, and we discuss everything in detail, right? Okay, we discussed that
Yes.
B cells are going to be transformed into plasma cells, and the plasma cells further are going to give you your antibodies, right? But the first
Yes.
antibodies which your B cells can give you only is your IgM. But if we
IgM,
need other kind of antibodies, for example, for your mucosal immunity, IgA, we need class switching.
class
And for the
switching.
class switching, we need CD40 ligand interaction. That's all. Okay, it was 53rd time, right? So we're going to repeat it 54th time for me. Yes, anyone?
I can try.
Hello?
Yes, I can try.
Yes, we understand.
Yes. You guys are open. You guys can just try.
Yes, doctor.
Yes, please.
We said we have B cells and T cells. The
Yes.
B cells act in the intracellular organism, for example, viruses.
The B cells?
The B cells.
Okay, anyone who can just correct
The
Dr. Margaret
intracellular viruses.
for the intracellular viruses, we are going to be having T cells, CD8 positive,
T cells. Okay, yeah,
presenting.
sorry, T
Okay.
cells. Yeah,
B cells
T
and
cells.
antibodies are for extracellular. Keep that in mind, right? Extracellular
Yes,
for B cells. Because antibodies cannot enter into the cell, right?
yes, into the cells. Okay. And then you said we have the MSC class 1 and MSC class 2. And the MSC class 1 is the CD8 that, show loves for the MSC class 1. And then the MSC class 1, I think the MSC class 1 is for the B cells, sorry. And then we have the cells that are outside with the antigen presenting cells. The antigen presenting cells is the MSC class 2, and the MSC class 2 react on the CD4. And in the CD4, they will have the T helper cell 1 and T helper cell 2. The T helper cell 1 is for macrophages. And for example, the macrophages can do granulomas, for TB. And then we'll have the T helper cell 2, which act on the B cells, and then the B cells can turn into plasma. And in the plasma, that's where we have the antibodies. And the first antibodies that is made is IgM. And sometimes when the IgM is made and then there's no class switching, we can have a condition that is called hyper IgM syndrome. And the IgM, they can do class switching. If they want to change to other immunoglobulin, they have to do what we call class switching to have E, D, and A, like immunoglobulins.
Okay, that's perfect, right? So, Dr. Moaz, hello. Okay, Dr. Moaz is also going to join the session. Okay, just give me a second. Okay. Okay, perfect. Right. So let's discuss our antibodies, right? So we have been, discussing the antibodies that B cells just give rise to the plasma cells. Plasma cells basically give you antibodies and everything, right? So let's discuss the structures of antibodies in a little bit detail, right? But before discussing antibodies, let me see if Dr. Moaz is, free, so he can just okay. Just give me a second. Okay, so let's discuss antibody structures in a little bit detail, right? Okay, okay. So, can you guys just see my this page of first aid? Okay, I'm going to make it really easy for you guys. Remember, right? So, we have two portions of your antibodies, right? Your Fab and your FC portion, right? So there you can guys can see it's a Y kind of structure. Y kind of structure. Your antibody basically is having this structure Y kind. So we have something called variable region, right? So to this variable region, basically our antigen is going to be attached. Our antigen get attached to this variable region, right? Then we have the constant region in our FC portion. So that just that isn't of significance. But here is a point which I want to mention is really important, right? Okay, remember every antibody is specific to specific antigen. Every antibody is specific to specific antigen. Is it clear? Hello? Okay.
Yes, yes.
Okay. So let me just give you example of that. For example, if you are buying something from a shop, right? You guys have seen there is a barcode on every product, right? And you have to scan that, and it's going to give you the price of that product, right? So every barcode is specific, right? Is it clear? Yes or no? Hello?
Yes.
Okay. For example, if we have an antigen, if we have a foreign body, it would be having a specific barcode, remember, right? For example, if that barcode is 312, let me just give it a random code 312, right? Let me just repeat it again. We have an antigen, it have a specific barcode, and that code is going to be 312. What's the code? Can anyone just repeat it? 312,
1,
right?
2.
Perfect.
312.
Okay. Remember, this variable site basically is having three genes. One, two, three genes. Okay, one, two, and three genes. One, two, and three genes. For example, if a pathogen or if an antigen from outside come into your body and it is having three, one, two. So what your variable region is going to do? It already have three, one, two, three on its surface, right? So it would just start keep on building, random numbers. It would just keep on making random sequences of 1 2 3, and in the end, after making millions of sequence, in the end he has some chances to get that 312. Is it clear? Yes or no?
Sir, can you please repeat?
Okay, let me repeat it. We have an antigen, it have a barcode of 312, right? Inside this variable region, we have some genes. Okay, we have three genes, one, two, and three genes. Let's suppose, right? I just want to make your concept one, two, and three genes. Gene one, gene two, and gene three, right? In order to fight off a pathogen with 312 gene, in order to fight off a pathogen with a code of 312 gene, what your variable region is going to do? It's going to start making random sequences like 1 1 2 2 2 1 2 1 2, and in the end, after making a million of sequences, he's going to make one of the sequence that is 312. So now this 312 in this variable region is going to match that pathogen 312. So this antibody is going to be specifically towards that antigen. That's why we say that every antigen or every antibody have specific antigen. Is it clear? Yes or no?
Yes.
Okay. So this
Yes.
1 2 3 was basically your VDJ gene, right? VDJ gene. So VDJ is going to, have a random combinations until he get, the combination which is specific for a specific antigen. Is that clear? Yes or no?
Yes.
Okay, perfect. That's fine. So we have two regions of our antibody, right? FAB region and the FC region, right? So the FA region, you can see that in the FAB region, basically, okay, just give me a second. Okay. So in the FAB region, here you guys can see that we have this light chain. Okay, we discussed that we're going to be having this antigen binding variable, region. After that, we have your FC portion. So the FC portion have something called the constant region. The constant region here, you can see the carboxyl terminal that aren't like that high G, right? If you just skip it, you won't see any MCQ related to that. Okay, but one thing which you should just keep in mind, the FC portion is really important for a process, that is your opsonization. You know what is opsonization? Yes or no?
Engulfing bacteria or
Yes.
pathogen.
Sorry. Hello, can you just repeat
Engulfing
that?
any pathogen.
Okay, directing any
Make
pathogen.
test.
If you are directing any pathogen towards certain area, that's basically known as opsonization. Like in our complement system, we are going to discuss certain opsonins, right? That is the C3b component. We are going to discuss that tomorrow in our complement system lecture, right? Okay, maybe Dr. Moaz just takes sessions today as well, right? So he will be taking session because it isn't confirmed, but he just told me that he can just take session after Dr. Moaz session. Okay, so what we were discussing is that generation of antibodies. We know the VDJ concept. Okay, so the random addition of nucleotide is going to occur until we get that specific, that specific kind of code which basically matches that specific antigen. Okay, we are also going to be having something called somatic hypermutation and affinity maturation. Do you guys have any idea what is affinity maturation? Hello,
Did you say affinity?
affinity maturation.
Okay.
No, sir.
Okay, that's really easy.
Exciting more interest.
Perfect. More affinity, more love, right? So here in the variable region, you can see in the variable region, once an antigen is attached there to the variable region, right? Those antigen who have more affinity towards that variable region would survive. Those antigen which have more affinity towards that variable region, as we discussed the concept of VDJ recombination, right? So the 312 and 312 are going to be having more affinity, right? Because their codes are same, right? So the hypermutation is going to occur in order to give you affinity maturation. If we have that same code, we'll be having more affinity between these, the antigen and the variable region. And in that way, those and those antibodies are going to be loving one another due to same codes. That's all. That's your affinity maturation, right? But if we do not have proper love or affinity there in the variable region of your antibodies, we they won't be having affinity and they won't undergo their proper function of destroying that pathogen. That's all. Is it clear?
Yes.
Okay. Perfect. So let's discuss our last topic for today, that is immunoglobulin isotypes, right? So just give me a second. It's really important. So tomorrow we are going to discuss the complement system. Along with that, I'm going to discuss some pathologies. Okay. So can you guys just see my screen? The immunoglobulins
Yes.
isotype?
5 types.
Okay. Perfect. Immunoglobulins isotype. We discussed. We already know everything, right? We discussed that the first antibody is made by your B cell is going to be your IgM, right?
IgM.
Okay.
Yes.
Remember, IgM is the biggest of all. It's the biggest of all. It's a pentamer. You guys can see 5 antibodies when combined with one another via a chain known as J chain, right? They're going to make this IgM,
Yes.
this big IgM. It's a pentamer. It's a big structure. It's a pentamer, right? So what do you think? Can this big structure cross placenta? Yes or no?
No.
No. Perfect. Perfect. Okay. So let's discuss the concept of avidity and affinity. That's really important. You guys are going to be having a Google MCQ related to that, right? Remember, we discussed affinity. Where do we see affinity? Where do we find affinity? Affinity can be seen in that variable region of antibodies. Am I right? Yes or no? Let me just zoom in this picture.
Yes.
You know, if an antigen is attach here to the variable region tightly or strongly, we are going to be saying that they have more affinity, they have more love. Yes or no?
Yes, yes.
Right?
Yes.
Perfect. Perfect. But the avidity basically means that how many numbers of antigens are binding to a specific antibody. How many numbers? You know? So looking towards all of the structures of these antibodies, you guys can see the biggest of all is your IgM. Am I right?
Yes. Yes.
Right?
Yes.
So the IgM can get,
Yes.
okay, there are 5 into 2, 10. They have 10 sites for the antigens to attach. Can I say that?
Yes.
Okay. So can I say that the avidity of IgM is highest of all? Because the avidity
No.
I just told you is the number of sites where your antigens can attach. Can I say that?
Yes.
But the
Yes.
affinity of IgM is not good. Why? That's very simple. For example, if one of the antigen attached to this variable site in IgM, the second one attached. So you guys can see that as we are attaching more and more antigen to these sites, we are overcrowding this IgM. We are overcrowding this IgM. So the crowd is going to be so more that if you are just adding some other antigen, it would be difficult for them to come into this crowd, right? Because they're going to become more crowded, you know. So keep in mind the affinity of IgM is the least, but avidity of IgM is maximum. Avidity is the number of sites where your antigen can bind, and affinity is the love of the individual site for individual antigen. Is it clear? I'm just making these definitions so that everything is clear. Is it
Yes.
clear? Yes or no? Okay, perfect. So, okay, then we have our IgG. IgG is the simple one, just a Y kind of structure. You guys can see only two sides. If its affinity is more, but avidity is less, right? Guys now know the concept. Okay,
Yes.
perfect. Remember, IgG, can it cross placenta? Yes or no? It's small.
Yes.
It's the smallest of all. Perfect. So
Yes.
it can
Yes.
Agree.
cross placenta, right? So in our hematology section, we are going to discuss a lot of pathologies, right? So sometimes a baby with autoimmune disease can be, he won't be having any symptoms for his first 6 months. Why? Because for the first 6 months, that IgG which is coming from the mother into the baby is gonna give this child immunity, right? But
Yeah.
once these IgG levels wanes out, then the baby is going to be having problems, right? Then this baby is going to have a lot of problems. So it means that IgG can cross placenta. If it can cross placenta, it means it's going to provide immunity for the first 6 months, for the child first 6 months, right? Okay. So we are going to be discussing something called autoimmune hemolytic anemias, right? Okay. As the name indicates, what is your autoimmune hemolytic anemia? Hemolysis due to autoimmunity. Okay. Or what do I mean by autoimmunity? Your immune cells destroying your red blood cells. In a simpler word, would be your autoimmune hemolytic anemia, right?
Yes.
Okay. Perfect. So, what happens in this case, the IgG, that in autoimmune hemolytic anemia, we can have two variants, right? What are those variants? We can have
Warm and
the
cold.
first variant, that is hot and cold, right? So the cold autoimmune hemolytic anemia is going to be seen in the peripheries, right? In the peripheries, in the extremities, in the hands, in the feet, right? While the hot one is going to be more centrally, because you can simply imagine that peripheries are cold while your central body is hot, right? So you have
Yes.
cold and hot, cold and hot. Peripheries are cold, your central body is hot, right? Okay, or
Yes.
warm. Okay, warm or hot. Same. Those are same things, right? Okay. So we can have, okay. So the warm variant of autoimmune hemolytic anemia involves IgG killing your red blood cells. IgG killing your red blood cells. And the cold variant of autoimmune hemolytic anemia involves, yes, IgM killing your cells. That's all. We are going to discuss it in detail in our hematology section. But it was just a brief overview, okay? Don't worry. Is it clear? Hello?
Ye-
Hello?
Again,
Again?
can you explain it again?
Okay. Okay. It's going to be a topic of our hematology, right? But let me just explain it. No problem. What is autoimmune hemolytic anemia? Anyone? What does the name indicate? Autoimmune hemolytic
Our own
anemia.
antibodies
Immune cell,
against the
immune cell will go
our
and kill
own.
RBCs.
Perfect. Autoimmune, our own immunity, killing your red blood cells. If they are killing your red blood cells, that's hemolysis. Hemolysis means breakdown of your red blood cells, right? So which component of immunity are going to kill? Your IgG and IgM can do that. So there are two variants of autoimmune hemolytic anemia, the cold variant and the hot variant. The cold variant is IgM
I
destroying your red blood cells in the peripheries of your body, in your hands, in your feet. Because hands and feet are cold as compared to your frontal body part, so there, the cold one is going to occur in the peripheries. While the hot is, I'm just calling it hot, but that's warm, right? Okay. So the hot variant of autoimmune hemolytic anemia is IgG destroying your red blood cell, but centrally in the warm area, right? In the hot area. Is it clear?
IgG destroying red blood cell, IgM destroying red blood cell.
Both of them are destroying the red blood cells, but the cold one is in the peripheries, and the IgM is basically doing that, and the hot one is basically in the middle of your body, your central body, and IgG are responsible for that hemolysis. That's all. That's all. Okay.
okay.
Hello. Okay. So let's discuss IgA. We know IgA. What is IgA? It's an antibody. What's the function of IgA? We know it's going to provide immunity to your mucosal surfaces. What are
Mucosal.
your mucosal surfaces? They can be your
And
GI
eye,
tract. It can be your eye.
respiratory
Every
system,
yes,
Saliva.
everywhere we have mucosa, right? Your saliva, secretions, breast milk contain IgA, everything, right?
respiratory tract.
Okay, respiratory tract, perfect. That's also your mucosal surface. Your secretions here, if you are just hearing, you're losing IgA. Just keep that in mind. Salivation, mucus, right? Okay. So remember, IgA. We know that normally B cells make IgM. In order to make IgA, we need class switching. Yes or no?
Yes.
Okay.
Yes.
So, if you remember, we have secondary lymph node organs known as Peyer's patches, right? What do we mean by Peyer's patches as secondary lymph node organ? It's the same lymph node tissue which we discussed yesterday, same lymph node tissue present in your ileum, right? In the ileum, you have this
Yes.
tissue, you have a tissue, lymphoid tissue, and
Pair
when
patch.
it they're going to be called as Peyer's patches. If it's a lymphoid tissue, it means it will be having follicles, follicles for your B cells, remember? Parafollicle T cells, right? So it means if it has follicles, it has B cells, so they have the ability to make IgA, right? Because B cells can be transformed into plasma cells. Plasma cells make your antibodies, IgA, via class switching CD40 ligand. Is it clear? Yes or no?
Yes.
Perfect, right? So all of the gut infections, for example, the giardia and everything requires immunity. The immunity is going to be provided by IgA. IgA comes from your biopatches. That's all. So let's continue our session of IgD, IgE tomorrow. That's all. The time is finished. Hope so you guys enjoy the session with me. You guys enjoy the session?
Yes, we enjoy.
Yes.
Oh,
Sorry,
thank you
sir,
so
I
much.
have a question.
Yes,
Yes,
please.
sir.
From where we
Yes.
can find the uh, these topics? I mean,
So,
recording of these topics.
so the recordings are going to be shared in the group, right?
Okay,
So
and the second generation of the group. So in there, I can't find the yesterday's session link.
okay, so you can just contact me. I'll just provide you the sessions. No worries. Okay.
Okay, thank you.
Okay. Please share the recording. Okay, for the recordings, I kindly request Dr. Hamza to share the recordings. It's Dr. Hamza's job to share that, right? I'll just try my best to provide, but I'm very weak at these things, providing the recordings. I personally do not even know how to operate a computer, right? Really sorry for that.
Okay, thank you so much.
Okay. Thank you so much. Thank you. Have a nice day.
Thank you.
Thank you.
Okay.
Excuse me, this class is finished?
Yes, the class is finished. Yes, everybody is supposed to leave.
Oh, so what's the next? Dr. Muaz will be presenting?
I'm really not sure. I think maybe tomorrow. Not sure about today.
Okay, thank you.
Thank you.